Hi, I'm Jack. I'm a third year Bachelor of Philosophy Honours Science student at ANU studying biochemistry. Today I'm going to be showing you how to do the autoimmunity probability experiment. Autoimmune disease occurs when the immune system, which normally protects us from nasty diseases, gets confused and starts to attack the body instead of bacteria or viruses. In this experiment, we're going to demonstrate how random chance can determine if someone develops an autoimmune disease such as type 1 diabetes or rheumatoid arthritis. We'll also see how genetics can determine if somebody develops a condition like this. Normally, the warriors of the immune system, the white blood cells, specifically B cells, produce antibodies against pathogens. Pathogens are microorganisms that cause disease, like bacteria or viruses. For example, the coronavirus that causes COVID is a pathogen. Antibodies bind to the pathogen, and this allows other cells to find the pathogens and destroy them. These B cells remember what these pathogens look like, and they keep you safe if you ever pick up the pathogen again, as they can produce antibodies even faster, meaning you don't even get sick. This is how vaccines work. They tell your body what to look for so it can produce antibodies faster the second time around and prevent you getting ill. Sometimes this process can go wrong, and the B cells produce antibodies against human cells. Normally, the body has mechanisms that can tell the difference between self and non-self cells, but this doesn't always work. Sometimes pathogens can even pretend to look like bits of human cells and trick the B cells into attacking the body instead of the pathogens. There are normally safety mechanisms that prevent antibodies that bind to human cells being produced, and a person then doesn't develop an autoimmune condition, but sometimes these don't work. For example, in type 1 diabetes, the immune system produces antibodies against the cells that make insulin, and the other immune cells then attack them. This is why people with type 1 diabetes have to inject themselves with insulin, as they can't make it themselves. In this experiment, we'll model the main checkpoints that the body goes through that normally prevent autoimmunity and see how, if they don't work, someone will develop an autoimmune condition. So checkpoint 1 is sometimes white blood cells, which are made in the bone marrow, or T cells, which are made in the thymus, have autoimmunity, meaning that their antibodies will bind to human cells. The body normally destroys them before they escape from the bone marrow or the thymus, and this stops them from causing autoimmune disease. Checkpoint 2 is right before an immune response is triggered to fight an infection. Certain white blood cells, called T regulatory cells, make sure that other white blood cells are not accidentally attacking the body's own cells. They have to do their job correctly to prevent autoimmune disease from happening. Checkpoint 3 is after a pathogen is destroyed by the body, white blood cells, specifically activated T cells and B cells, destroy themselves through a process called apoptosis to stop the immune response, as this extra immune response could lead to an autoimmune disease. So in this experiment, we're going to use multicolored sticks and a 6-sided dice to model these checkpoints and see how random chance can affect whether or not those checkpoints are passed successfully. So now let's actually do the experiment. So first, we're going to draw up our tables in which we're going to record our results. So each table is going to represent a group of people, hopefully at least 20 or more. The more people, the better, as that's more accurate and the model will better represent what's really going on. In each table, you need to make sure that there's enough columns to represent 20 people, or if you're doing it individually as part of a large group, you could do 1 or 2 people, your friend could do another 2, and so on, so that as a class, you manage to get the whole 20-person group represented. So you're going to label you're going to have 4 tables, one which is a population normal risk, a second one which is a higher risk at 1 checkpoint, a third one which is a higher risk at 2 checkpoints, and a final one which is a higher risk at 3 checkpoints. So these groups represent people with different genetic predispositions to autoimmune disease. So to do this, we're going to use coloured sticks, and so you're going to need to use the same number of sticks for each colour so that the chance of drawing a stick of each colour is exactly the same. So I'm going to use 3 sticks of each colour. Okay, so now we have 3 sticks of each colour in the bowl. And it's important to use the same number of each. It could be 7 of each colour, 5 of each colour, it really doesn't matter. As long as you've got the same numbers of sticks, then you're going to be representing the probability accurately. So once you've got that ready, we're now going to start playing the actual game. So first, you're going to draw randomly a stick from the bowl, and that is going to represent the self color of the first person that you're testing. So look away from the bowl, don't cheat. You need to make sure it's really, really random. So I'm going to look away and then I'm going to draw a stick from the bowl. Right, so I picked red. So that person's self color is going to be red. So I've written that in the table in column 1 under setup color. And now I'm going to do checkpoint 1. So checkpoint 1, if you remember, represents those B and T cells in the bone marrow and in the thymus that might have produced self-antibodies, and it's the body trying to prevent those antibodies from escaping into the rest of the blood system. So I'm going to randomly draw a stick again, so I'm going to look away. So it is red, so this means that those antibodies are going to recognize that person's cells. And so I'm going to write that the color does match. I'm going to write yes. And now, because it did match, we're going to roll the dice. So if it's a 1, 2, or a 3, that's a low number, we're going to say that the body has successfully stopped these autoimmune cells from escaping. If it's a 4, 5, or a 6, we're going to say that the checkpoint's failed and that the autoimmune cells have escaped into the body and the person will develop an autoimmune disease. So I'm going to roll the dice now. So it's a 6. So unfortunately, the body hasn't been able to stop the B and T cells from escaping. And so this person is probably going to develop an autoimmune disease. But still, we're going to move on to checkpoint 2 and see if the person will develop an autoimmune disease in a different way. So I'm going to put the sticks back. So now we're going to move on to checkpoint 2 to see if the body's T regulatory cells can prevent an autoimmune response. So I'm going to draw a stick randomly from the bowl again. Okay, so this time it's blue. So that doesn't match the person's self color. So I'm going to write no in the table. And so this time, because it doesn't match, we're not going to roll the dice. Okay, so now I'm going to put that back in the bowl and we're going to move to checkpoint 3. So this is after the body has acquired an infection, do those T cells successfully undergo apoptosis and stop producing an immune response? So I'm going to draw again randomly from the bowl. So this time it's yellow, it doesn't match. I'm going to write no, and we're not going to roll the dice. So then on the last row of the table, we've got, did this person develop an autoimmune disease? And so because in checkpoint 1, it matched, and then the number was above a 3, we're going to write yes. And it's important that you record all this data for every single person. So that's just person 1, and then I would do person 2, person 3, person 4, and you want to do as many people as possible to try and get the most representative sample. So that was for the normal risk group. And now I'm going to demonstrate how we might go about doing it for a higher risk group. I'm going to do this in the same table, but normally you guys would do it in your higher risk at 1 checkpoint table, higher risk at 2 checkpoints, or higher risk at 3 checkpoints table. So I'm going to do person 2, and we're going to say that they're a higher risk at 1 checkpoint person. So this high risk is because they've got a difference in their genes, which means that their body is not quite as good at regulating these autoimmune cells. So we're going to put all the sticks back in the bowl and we're going to draw a setup color again to see what the self color of this person is. So I'm going to draw it randomly from the bowl. So this person is going to be yellow. I'm going to write that in the table. And now we're going to put it back and we're going to go and do checkpoint 1 again and see if the body can stop the B and T cells escaping the bone marrow. So I'm going to draw it randomly. Okay, so it's a purple, it doesn't match, so I'm going to write no. I'm going to put that back. I'm going to do it again for checkpoint 2. It's a red, so I'm going to write no, and then I'm going to do it again for checkpoint 3. Okay, so this time it is yellow, and this means that it's matched again. And this person we're going to say is a high risk at 1 checkpoint. So because it has matched, we're going to write yes. And now, because they're at a higher risk, instead of 1 to 3 being low risk, we're only going to say that 1 is low risk. So any number other than 1 means that this person is going to develop an autoimmune disease. So for this person, we've chosen to do this at checkpoint 3, but you could do this at checkpoint 1, checkpoint 2, or checkpoint 3 if they're a 1 checkpoint high risk person. If they're a 2 checkpoint high risk person, then you're going to need to do it in at least 2 checkpoints. So that could be 1 and 3, that could be 2 and 3, that could be 1 and 2. It doesn't matter on the combination as long as you're consistent. And then the high risk at 3, we're going to do all 3 where only 1 means that they don't get the autoimmune disease. So I'm going to roll the dice now. So it's a 1. So this person's got really lucky. Despite their higher risk of getting a genetic autoimmune disease, their body has luckily been able to fight and stop that from happening. And so we're going to write no. Okay, so once we've done this for all different risk groups and we've got a full data set and we've got all 20 people, we're then going to start analyzing our results. So what we're now going to need to do is collate all the results from your class, and we're going to count up for every group, for the population group, for the high risk at 1 checkpoint group, for the high risk at 2, and so on. You're going to count all the people that did and all the people that didn't. And then you're going to try and work out the percentage of people that did develop an autoimmune disease and work out the percentage of people that didn't develop an autoimmune disease. So you can write this down in the table that we've given you. And then if you want to do more to understand how the risk varies depending on your genetic risk, you can put this into a graph. And that might make it easier to visualize how your genetic risk means that more people end up with an autoimmune disease. If you're really interested, you can go online and Google autoimmune diseases and try and understand why they happen and which different genetic differences put someone at higher risk. And it's really important that we have more people studying these things, because right here at ANU, we've got one of the world-leading facilities to study autoimmune diseases, and we're starting to finally understand some of the really, really challenging ones, like lupus and type 1 diabetes. I hope you guys really enjoyed this experiment. I certainly did, and I didn't get to do all of it, but hopefully you will, and you'll get through it all, and you'll get some really interesting data. This type of thing, modeling an experimental system, is really important in science. It may not be exactly what happens in the body, but it can tell us some really important information and help direct us in our next experiments.